9 years ago, I found myself in a doctor's office contemplating the nature versus nurture debate from a fresh perspective. You see, I had been trained as a geneticist and had spent my career manipulating DNA and seeing the profound consequences in a lab setting, so I'd always put my money more on the nature or the genetics side of the debate. But as my doctor revealed to me that I was pregnant with identical twins, I realized that my convictions were about to be put to the test. For starters, we had not budgeted on 2 daycare bills at once. So I kind of half-jokingly started to wonder what would be the consequences maybe if we just sent one twin to daycare and maybe just kind of tucked the other one in my office drawer during the workday. Despite their identical DNA, I somehow doubted that things would turn out all that well for the twin in the office drawer. Identical twins have had a profound impact on scientists' understanding of nature and nurture. Studies on identical twins who were separated at birth and raised in separate households have helped us understand different traits that are more affected by nature, or DNA, versus nurture, or the home environment. For example, some traits like IQ or criminal tendencies are more affected by your DNA than the house that you grew up in. On the other hand, other traits like depression in men or your preference for a particular political party are more influenced by your environment than by your genes. So what about identical twins who are raised in the same home environment? Their nature and their nurture are almost the same. And yet, any parent of identical twins, myself included, can quickly point out differences in their children. One twin may have more of a preference for certain types of foods, or may have more aptitude for a certain sport or a musical instrument. And sometimes, health differences can arise in these children. For example, there are reports of autism or asthma or bipolar disorder arising in one twin at a young age while the other one remains unaffected. How do we explain these differences given that the DNA is the same in these children, and for the large part, their home environment has been the same too? Well, it turns out that some of these differences can be explained by a third, very powerful influence on our lives besides nature and nurture, and this is epigenetics. I'm going to talk to you today about what epigenetics is and how it impacts your life, even if you're not an identical twin. So before we talk about epigenetics, we need to consider our DNA and how it fits into our cells. Because believe it or not, of the 50 trillion or so cells in your body, each one contains about 6 linear feet of DNA. I mean, if we were to stretch it out, it'd be about as tall as a pretty tall man. So how in the world do we fit that amount of genetic material into something the size of a cell nucleus, which is 400,000 times smaller? Well, the answer is that we do it by wrapping our DNA around clusters of proteins called histones. You can think of histones sort of like molecular spools, and there are about 30 million of these spools in each of your cells. So this helps explain how you fit such a tremendous amount of DNA into a small space. Now, we call this combination of histones and DNA chromatin. And while chromatin solves this tremendous packaging problem that the cell has, it also presents a new one for the cell. And this is one of DNA accessibility. Because keep in mind that the functional units of DNA are actually the genes encoded in it. These are the instructions for the cell. These are what tell the cell what to do and who to become. And yet, when these genes are tightly compacted into a chromatin structure, the cell is unable to read them. They might as well not even be there. And this is where epigenetics comes in. So epi, meaning on top of, in genetics, your genes, literally refers to a set of instructions that sits down on top of our DNA and our histones. Epigenetic marks are small chemical tags which sit down on our chromatin and can help instruct it whether to compact or decompact. And those instructions can then affect how the cell reads the underlying genes encoded in the DNA. So to show this schematically, some epigenetic marks, shown here in red, can help condense chromatin. And when they do this, they obscure the underlying genes, preventing the cell from being able to read them. They turn those genes off. Other epigenetic marks, shown here in green, can help decondense the chromatin. And when they do this, the gene becomes accessible to the cell. The cell is able to read it and turn it on. Now, these types of epigenetic marks are profoundly influential to our biology. Consider, for example, what is it that makes our cells different from one another? What makes them look and behave differently? What is it that makes a muscle cell, for instance, look different from a neuron? After all, these cells contain exactly the same DNA, but it's their epigenetic instructions that help tell them which genes to turn on and which ones to turn off. And with those different genes at play, these can become very different cells. So you might be wondering, when does all this epigenetic information get laid down on our chromatin? And the answer is that much of it happens during our embryonic development. So interestingly, when you were first conceived and you were just comprised of a few undifferentiated embryonic stem cells, which had the potential to become any cell in your body, your chromatin didn't have many epigenetic marks on it. It was only as your cells began to divide and receive signals and information from surrounding cells that the epigenetic marks began to accumulate, and then the genes began to get turned off and turned on, and the muscle cell became very different from the neuron. This brings me to a really important point about epigenetics, and that is that epigenetic marks can be influenced by the environment. And when I say environment, I don't just mean those surrounding cells that tell a neuron to become a neuron. I also mean the environment outside of the developing embryo. So the food that mom eats, or the prenatal vitamins that she takes, or the cigarettes that she smokes, or the stresses that she encounters at home or at work, can all be transmitted as chemical signals through her bloodstream to her developing fetus, where they can get laid down as epigenetic marks that affect the fetus's own genes